Hello everyone, and welcome to today's live broadcast, Pediatric Cancer Genomics, Accessing Data, Tools, and Visualizations in St. Jude Cloud. The team from St. Jude Children's Research Hospital will be the speakers for this event. We are excited to bring you this educational webinar presented by LabRoots, the leading scientific social networking site and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Microsoft, a leading cloud solutions provider empowering health organizations and patient care in DNA Nexus, the leader in cloud-based genome informatics and data management. The DNA Nexus and Microsoft partnership enables the advancement of genomic discovery through the sharing of tools and data sets in a secure, compliant, and collaborative ecosystem. Our work accelerates scientific insights in healthcare and research to solve the largest challenges in the market today. I'm Singer Ma, the Scientific Operations Director at DNA Nexus, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the QA button located in the bottom of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you're having any problems seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. We'll start this webinar with a quick introduction to the infrastructure that powers the St. Jude Cloud. Developed in partnership between St. Jude, Microsoft, and DNA Nexus, St. Jude Cloud provides a flexible cloud platform for rapid data mining, analysis, and visualization capabilities. The St. Jude Cloud Platform was built on top of DNA Nexus and runs on Microsoft Azure. It is a customized view and request system for St. Jude data and offers the use of St. Jude tools. St. Jude Cloud Platform seeks not only to provide a valuable data set, but also the cloud platform to allow in-place analysis of the data. Too many of the existing data access platforms simply provide data and tools to download them. However, this doesn't solve the core problem of having multiple copies of data, and as data volumes grow, this cost increases. St. Jude Cloud Platform is more than just an FTP server, providing the underlying framework to perform novel analysis. The system also allows for easy running of the St. Jude tools on data which the user brings in. Everybody has had the experience of trying to use existing bioinformatics tools. You'll go to a website, download a zip file, then go through the arduous process of installing things onto your system. The St. Jude Cloud Platform seeks to simplify this whole process. All it takes is uploading your own data, choosing which tool you would like to use, and running it on DNA Nexus. All of this is made possible by the secure and compliant system created by DNA Nexus, allowing St. Jude to breathe easy and not worry about these issues. Now, I'd like to introduce today's speakers, the team from St. Jude Children's Research Hospital, who will share insight into how they built the world's largest public repository of pediatric cancer genomics data. We have Scott Newman, Ph.D., Group Lead in Bioinformatics Analyst, and Clay McLeod, Manager of Bioinfo Software Development. Please join me in welcoming the first speaker, Scott Newman. I will now turn the presentation over to him.
Hi there, everybody. Nice to meet you. My name's Scott Newman. I'm a Group Lead of Bioinformatics Analysis within the Department of Computational Biology at St. Jude Children's Research Hospital. And later on, you'll be hearing from my colleague, Clay McLeod, Manager on the Cloud Project. But first up, we have nothing to disclose. We have no financial relationships with companies that we will mention. In this webinar, we aim to introduce you to the St. Jude Cloud. What is St. Jude Cloud? Show you how you can access data, show you how you can run St. Jude tools on your own data, show you briefly how to port your own tools into the St. Jude Cloud ecosystem to analyze data, and lastly, show you how to use some of the visualizations to gain insights from the vast amount of data that we're offering freely to the public. So, driving all of this is our desire to share data with the global research community. St. Jude is committed to sharing that data as we're committed to finding cures for pediatric cancer and other rare and catastrophic diseases of childhood. We were among the first to start sequencing large numbers of whole genomes and whole transcriptomes with the Pediatric Cancer Genome Project, which began almost 10 years ago. Since then, we've done a lot more sequencing, namely with the three studies that I list here: the Pediatric Cancer Genome Project, the Genomes for Kids study, where we took whole genome sequencing into the clinic, and the St. Jude Lifetime cohort, where we followed thousands of long-term cancer survivors and also performed whole genome sequencing on them. As these studies have been published, we've always shared the data with EGA and dbGaP and other repositories, and it's been possible for researchers worldwide to access the data through a data access committee and then download the data onto their local infrastructure, and they can run their analysis. And that's been fine for the past few years. But now that we're generating hundreds and thousands of whole genome sequences, the data is getting so big that the old model of data download is becoming challenging. And it also assumes that you have sufficient infrastructure at your institution to be able to house and analyze this data. A few years ago, we started thinking that there was a better way to do this. What if you were to come to the data? What if you were to bring your tools to the data? And with that idea, the St. Jude Cloud was born. It's taken 3 years now to get to where we are. And early on, we considered the other cloud computing options out there and decided whatever we came up with had to be secure, it had to be sustainable, and it had to be expandable. We're now generating thousands of whole genome sequences every year, and we want to share those with the global research community. But to get there, we had to get some help from the outside. First up in 2016, we selected a software development partner, and we went with DNA Nexus because their platform was highly secure. We found that it was easy to develop on. We could move our tools to the DNA Nexus infrastructure relatively easily. And most importantly, they were excited to work with us. Next, in 2017, we partnered with Microsoft. There are costs associated with cloud computing, and storing all of these whole genome sequences, terabytes and nearly a petabyte of data currently we have, would cost a lot of money were it not for the philanthropic gesture from Microsoft. They're housing all of our whole genome data from the studies I mentioned earlier at no cost within Microsoft Azure. And between them, the partnership between St. Jude, DNA Nexus, and Microsoft made the St. Jude Cloud possible. We launched in April of 2018 with our first 5,000 whole genome sequences that we can offer to the global research community. And we've been fulfilling data requests from April to now September. So what does it look like? Here is the St. Jude Cloud, www.stjude.cloud. Hopefully that web address is easy to remember. The website is split into 3 sections: data, tools, and visualizations. I'm a scientist. I'm a self-taught programmer. So really what I want to do is get my hands on the data, run some sort of analysis tool on it, and look at the output. I think many of us in the bioinformatics and computational biology field can relate to that workflow. And what I'm going to do in the subsequent slides, and with Clay's help, is walk you through these 3 sections and show you how you can gain access to the data, run the tools, and look at the visualizations within St. Jude Cloud Platform. So first up is data. These numbers are, as of April 2018, we are currently undergoing a big push to get up to 10,000 whole genome sequences by the end of 2018. Currently, there are over 5,000 whole genome sequences, over 5,000 exomes, and over 1,200 RNA sequences. These come from common and rare pediatric tumors, as well as matched normal tissue from the tumor donors. All of these samples have been consented for research data sharing. Importantly, the data set is also harmonized. We remapped everything to the latest build of the human genome, GRCH38, using the Microsoft Genomics service GATK best practices pipeline. As I just said, the data is de-identified and it is consented. However, this is genomics data, so we're not giving it out for free, and well, free is maybe the wrong word. The data access is protected. We go through a data access committee if you wish to gain access. You can select broadly based on study, for example, the St. Jude Life, the Pediatric Cancer Genome Project, Genomes for Kids, or our clinical pilot study. But we've also tried to make it easy to drill down to specific samples of interest. Using our PCAN visualization portal, you can identify the samples that you're interested in, for example, high expressors of JAK2, and put them in your shopping cart and submit that request to St. Jude Cloud. So here's what I was alluding to earlier. You must apply for access to raw data. It is pediatric genomics data, but we've done our best to streamline the data access procedure. All you need to do is download a form and sign a legally binding agreement. If you don't want to download the data, if you want to work with it exclusively in the cloud, then you're good to go. All you need to do is upload the form, and that form will be forwarded to our data access committee, and hopefully they'll be able to grant you access quickly. If you do wish to download it, you need to get somebody from your institution to sign off that you have appropriate data security procedures in place and the requisite amount of storage. Now, I realize that was a lot of information, so at this point, I'm going to hand it over to my colleague, Clay McLeod, and he's going to walk you through the data identification, request, and access procedure.
Okay, thanks, Scott. So what I'm going to do is walk through the data section here first. So if you guys can see my screen, I'm on stjude.cloud right now. This is the home page. So going in, and I'll click on data here. We've got, if you've made previous requests, you can click here to go and look at those previous requests, but we're going to go in and explore the data first. So this is the part of the site that we call the data browser. So there are three main ways by which you can choose what samples you want by the primary diagnosis here, which has all sorts of different information and diseases, by the publication. So this ties up with, different PubMed IDs. And then also by this general purpose data set tab. So here are the data sets that Scott's been talking about here. So we've got all of the data from the Pediatric Cancer Genome Project. We've got the data from the clinical pilot cohort where we piloted whole genome, whole exome, and transcriptome sequencing in the clinic. We've got the continuation of that, which is the Genomes for Kids project, and then all of the cancer survivorship study stuff under the St. Jude Life data set. So the process is relatively simple. You just tell us what samples you want here. After you tell us what samples you want, you tell us what type of files you want from those samples. And then you click Request Data. Now, on my screen here, I actually already have access to all of these. So since I was approved for access previously, I had submitted the data access request. I could just get the data now. I don't have to request access. If you haven't been through that before, you will have to request access by downloading the PDF and everything. So I've already done this, though. I'm going to go through and I'll show you what that request looks like in a second. But first, I want to let me go back here really quick to data, and I want to show you how to select data using PCAN. So PCAN is the Pediatric Cancer Portal that we develop here at St. Jude. It's a lot of visualizations that I think Scott's going to talk about in a little bit. But you can do some really cool things here. So I'm going to go through and look at my favorite gene, JAK2. And I'm a researcher. This is the gene I'm interested in. So you can see that here are point mutations that we've seen in JAK2 in our pediatric cohort. We have a graph over here for RNA-seq expression. So I'm going to focus on this just for the purposes of this demonstration. And I'm going to do a log 10 transform here. So these are all of the samples that we've seen that are loaded into Pecan here. And we have low expressors of JAK2 all the way up to high expressors of JAK2. So let's say I'm a researcher, I want to look at all the raw data for the high expressors of JAK2 that St. Jude has. So what I would do is I would come in here and I would select these, right? And you can see a circle pop up here that shows me the breakdown of what are these samples, like what type of disease do they have, things like that. But actually something's interesting is down here, it says add 289 samples. So this is the add to cart functionality that Scott had talked about previously. You can click here and say, I want all of these samples to be added to my cart, click my cart, and then I could submit a request to St. Jude Cloud here for all of the samples that we have in St. Jude Cloud that match up in this cohort. So as you can see, it's not all of them today. There's 376 that I've selected here in Pecan, and only 289 of those are in the platform, but we're working to make that a one-to-one relationship. So I'm going to step back here and just show you the last bit, which is, what if I've already made a data request in St. Jude Cloud? So in that case, I would click Manage Data here. And I've already done some sample requests, so I'll show you the clinical. I went and checked out all of our clinical genomics data on St. Jude Cloud. So I click this, and what it's going to do is it's going to open up a new tab with my DNA Nexus St. Jude Cloud project loaded. Well, I'm logged in. Okay. So here is a DNA Nexus and a St. Jude Cloud branded DNA Nexus project, and we've got all of the BAMs are going to be in here. It might take quite some time to load because there are thousands and thousands of BAMs. 3,448 BAMs with the associated indices, and then we've also got GVCF files here. So that's an overview of data in St. Jude Cloud, and I think with that, I'm going to kick it back over to Scott to walk you through tools.
You're back with me, Scott. Moving on to the next section of the website, tools. Many of us in bioinformatics are frustrated at how difficult it is to install tools and get them up and running on your data. There's a big problem with reproducibility in bioinformatics, and we're all aware of it. We all read papers and we think, this tool is really cool, I want to run it on my data, I want to see what it gets, and then we waste days and months trying to install the tool, getting it to compile, getting appropriate libraries, getting whitelists, getting blacklists, and even then there's no guarantee that you can reproduce the data in the published study. We've tried to remove all of that pain by packaging our tools into workflows that are easy for a non-bioinformatics specialist to run. Somebody like a graduate student or a postdoc or a medical doctor can hopefully run our tools with only a few clicks of the mouse. So we've aimed to make these tools easy to use, and most of them also produce an interactive visualization at the end that you can explore. Currently on St. Jude Cloud, we have our rapid RNA-seq pipeline. This is a method to find fusion genes in cancers, and a similar method's being used in our clinical pipeline to support one of our clinical trials. We make this tool available for research use. We also have ChIP-seq broad and narrow peak calling, neoepitope prediction if you're interested in immuno-oncology. We have a differential gene expression pipeline called Warden, and we have our cancer predisposition variant annotation and classification system called PecanPy. Coming soon is going to be a tool that will allow you to unpick mutational signatures such as UV or tobacco smoke damage from cancer genomes, and that should be coming out in the next few months. As I said earlier, it's our hope that only a few clicks will allow a non-specialist to run these tools and get meaningful results out at the end. At this point, I'm going to hand it over to Clay again to demo the rapid RNA pipeline and actually how to get your own data into St. Jude Cloud.
Okay, so now we're going to take a look at tools. So as Scott was showing in the presentation, here's our list of the tools that we make available in St. Jude Cloud today. I'm going to walk you through Rapid RNA-seq, which Scott and I both worked on. So when you click on Rapid RNA-seq, you're presented with a view of, it's a profile view of this tool. We've got a nice icon up here, but more importantly, we've got a lot of information about the tool. So who wrote the tool, if there's a publication, the inputs, the outputs, the supported genome builds, and then a contact us button. There is extensive documentation. If I click here on getting started, you can see that we've got a page full of documentation telling you about how to run the tool, monitoring run progress, analysis of results, known issues, and frequently asked questions. You also, we can upload our data on this tool. So uploading data is pretty easy. We have an app called the St. Jude Cloud Data Transfer app that you can just point and click. So I'm going to click here to open app, and it's going to open up the St. Jude Cloud desktop data transfer app on my computer. You can see that it has preloaded here my Rapid RNA-seq project. This is the area where we're going to be running Rapid RNA-seq in the cloud. And uploading your data is pretty simple. Oops. Uploading your data is pretty simple. You just take the files on your desktop here, drag them over, and then you click upload. And those will be transported, beamed up to the cloud into your secure cloud environment. So that's how you upload data. I'm going to show you where that data went here in just a second. I'm back on this page, and I'm going to click Launch Tool. Select the preset. So I had uploaded, a pair of FASTQ files, so I'm going to select that. And I get dropped into my St. Jude branded DNA Nexus project with this workflow open. So you can see there's a lot of steps here, a lot of engineering work that went into this, but the key piece are these two orange boxes right here. So what you're going to do is point and click and hook up those two files that I've uploaded previously. So here I've got a folder called Uploads that I put these into. This is where things that are clicked and dragged and uploaded through the St. Jude Cloud Data Transfer app get uploaded to by default. So I'll hook up R to that first slot here, and I'll hook up the R2 to the second slot. And then I click Run. So that's all there is to it. I mean, we have a few parameters that you can adjust here, but we don't recommend, we have very same defaults, so we don't recommend that you go in there unless you followed the user guide, want to set something custom. So once you kick off a job, I've already done this previously, and you can go in here to the Monitor tab and see how are my jobs doing? Did they fail? Are they still running? How much did they cost? You can see this is very reasonably priced for calculating all of these gene fusions in an RNA-seq sample. And it looks like everything was good. If I really wanted to, I could click through and look at the logs for each of these runs, but for the purposes of this demo, I'm not going to do that. So your tool, you uploaded your data, you ran the tool, waited a few hours, and now it's done. So let's take a look at some of these results files. We've got a bigwig file that's output. We've got the aligned BAM file from STAR. We've got some fusions. I'm going to key us in on this one results file here, which is called the Diffusion Viewer. So all tools in St. Jude Cloud, we aim that there's a really nice interactive visualization that you can use to analyze your results rather than just a tab delimited text file or an Excel file. So let's load this up. So what I've got here is a view showing all of the putative fusions found in this RNA-seq sample. So if I click here on the results are sorted from what the tool thinks are the fusions that you should look at first, down to things that maybe you should read through, but the tool doesn't think are as important. So we have a bunch of high-quality in-frame fusions here, and for the astute among you, you'll notice that there's the BCR-ABL fusion here. And there's a nice view of the protein that's been formed here. I can toggle this chimeric view and show a nice, what the byproduct of this fusion is. Now, also you'll notice that the tyrosine kinase domain is preserved in this example. So it's really neat. It's a nice way to go through and look at the different fusions that are produced. So so with that, I think I will hand it off to Scott again, just to go through the visualizations in St. Jude Cloud.
You're back with me, Scott. So Clay just showed you how you can run a St. Jude tool on your own data. But how about if you want to run a novel analysis, something that we might not have a tool for, on our data, or indeed on your own data within the cloud ecosystem? The new mantra is becoming bring the tools to the data, because the data from whole genome sequencing is huge, whereas the tools are relatively small. And as I'll show you in the next couple of slides, it's actually quite easy to port your tool. I think a programmer of quite average abilities, such as myself, can quite easily do this. So here's a simple demonstration. Hopefully you've all heard of the popular RNA-seq quantification tool, HT-seq, published by Anders Pyle and Huber in 2015. What you do is give this tool a BAM file of mapped next-generation sequencing reads. You give it a GTF file that you can download from Ensemble or UCSC, which defines your gene model, and it will count the number of reads that overlap with your gene model features. So here's just a simple program that I've written that I run in our local compute cluster. On the first line, you define the name of your BAM file, testfile.bam. The next line, you derive the name of that file, BAM name. Then you define the name of your output, your BAM file name plus htseq.txt. And then you define your gene model, GTF, refseq.gtf. And you run the command htseq count. F, I'm giving it a BAM file. R, I'm telling it position. S, no, you don't have to worry about these parameters. And I pass it the BAM file and the GTF file, and it will output. This is probably the simplest bioinformatics program you'll ever see. But how do I move that from my local computer into the cloud? What you have to do is define your inputs and outputs in a JSON file, which is a popular format to transfer data on the web. You give your tool a name. You tell it what your BAM file is going to be called. You tell it what your GTF file is going to be called, just like I did previously. You tell it what your output file is going to be called. Then you load your GTF file into a secure one-off instance inside of DNA Nexus. You load your BAM file and your GTF file, and you run the exact same htseq command as you did before. You then output the data. So really all we're doing is wrapping a single line of code in the DNA Nexus framework. Now, for the non-programmers out there, you may find this to be quite challenging, and there is a little bit of a hump to get over. But I think you need a relatively low skill level to be able to do this. For the bioinformatics professionals who might be listening, I hope you'll agree that this is quite a straightforward procedure. Of course, what I've demonstrated is just the tip of the iceberg, but the outcome is actually you now have an htseq tool that you can deploy to the cloud using a simple dx build command. And it appears here in the St. Jude Cloud DNA Nexus style project as St. Jude htseq. You can then run the tool with a simple click. You no longer have to program at the command line. All you need to do is to have written the code on the previous page, and that tool is now there for you to use. The added bonus is you can now share that tool with your lab members or collaborators that might be anywhere in the world just by granting them access to your project. It's our hope that this system is simple enough for non-specialists to learn, and it should be easy enough for bioinformatics scientists to learn. There's lots and lots of other complexity that you may wish to introduce. For example, DNA Nexus is now offering common workflow language and workflow definition, CWL and WDL support, if you want to move pipelines from other platforms, as well as support for Docker containers, parallelization, etc. It's all in there and it's all very well documented. So that's all I want to say about tools. And lastly, you don't have to be a bioinformatics wizard to use this thing. Absolutely anybody can explore the data. The visualization sections of St. Jude Cloud has aimed to make simple, intuitive, and interactive visualizations that anybody who's interested in cancer genomics or some of the other diseases we study to make novel insights from the data visually and with their mouse. You don't need any programming skills. Many of these visualizations have been linked to high-profile publications, and we've made the data and the visualizations available. But I'd like to zoom in on our new one. This is a software we are calling Genome Paint, and it is a new genome visualization engine powered by Protein Paint and Shinjo's visualization team here at St. Jude. I'm going to hand it over to Clay, and he can walk you through P53 and some interesting mutations as visualized through Genome Paint.
All right. So last but not least, visualizations. So you can go to, you can click on the visualizations tab from the front page, and this is where it will land you. We have a bunch of different study level visualizations, which I'll get to in a second, but I'm going to key in on this PCGP somatic and germline mutations. So what we've done for the Pediatric Cancer Genome Project is we've taken all of the variants that we have for that, and we've put them into one view for you to kind of browse and see if any of the data here can help you in your own studies. So first up, we've got some ataxi tracks to hint at possible regulatory regions. We've got the copy number variants in PCGP. We've got some cosmic hotspot mutations, and then we've got PCGP, the somatic mutations, and then the germline variants called by the Microsoft Genomics service. So down here at the bottom, we have a very extensive filter view, all of the ways you might want to slice and dice this data. So what I'm going to do for the purposes of this demo is go through and filter only the pathogenic or likely pathogenic clinical significance, those that have been reviewed by our analysts. And you can see that there are many fewer variants here. So let's try this one. So you get a lot of information about this, and normally this is all you would see are these information, but if you apply for access, you can get down to the genotype of each individual patient. So that's, protected access, but in this case we have a sample here which in the germline sample in this graph, it has a variant allele frequency of probably about 0.35, and then you can see it also light up over here as it's up to around 0.75 up here. So in this case, you might examine this and say, hey, that looks similar to a loss of heterozygosity event in this sample, things like that. So there's a lot of really cool things you can do. I'd encourage you guys to go out there and explore this, give us your feedback, things that you would want to see in this. Additionally, we have these study level visualizations. So frequently when we publish papers, we will have a place to come and interactively view the data. So let's try this one. The recent study by Jing Wei's group. Right. So, so here we have, there's a lot to go into about what this can do. You can open up things about what kind of variants are here and what genes are affected in individuals, but I'm just going to click around and kind of show you that we group different mutations here based on the disease type, and it looks like the gene that's affected. There's a lot of cool things you can do. It's very interactive and sortable, so you can see me clicking on things and things sorting based on what the genes of interest. Often we have metadata at the top here about, sex or age at diagnosis or a number of other things. So again, I would encourage you to go to the visualizations page, check it out, see if there are any publications you're interested in, and let us know if you have feedback. So with that, I'll turn it over one last time to Scott to finish out the presentation. Scott.
I'm back. And so that's about everything that we wanted to demonstrate today. We really appreciate you taking the time to view our webinar. And I'd like to close by saying, what is the future of St. Jude Cloud? Well, as I heard recently, the genome sequencing business has entered the exponential phase. We're generating a whole load of whole genome, whole transcriptome data on clinical samples and research studies at St. Jude, and we hope to be adding those to St. Jude Cloud as the studies are published. We're on track for 10,000 whole genome sequences by 2019, and those will be publicly accessible through the data access procedure that I just outlined. We'll be adding basic clinical data and epigenetic data. Other things like gene expression and copy number profile are also coming soon. We'll also be expanding into new diseases. If you're at the American Society for Hematology, you may want to look out for our sickle cell study that will be added to St. Jude Cloud shortly. In terms of tools, we're going to be adding our structural and copy number whole genome sequencing pipelines, which we believe are among the best in the business. And we'll be improving the integration of our protein paint visualizations with user data, so you can generate the same visualizations that Clay showed with your own data at some point in the near future. Also, there will be several new types of visualizations, so watch this space. This has been a big and complex study, and lots of people have been involved, and I'd like to acknowledge them and thank them for their help. That's all from me. Thank you very much.
Thanks, Scott and Clay, for that wonderful presentation. We now have time for some Q&A. If you have a question you'd like to ask, please do so now. Click on the Q&A button at the bottom of the presentation window. Type your question to the box that appears on the screen and click the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, what additional tools are you looking to add to the existing library on the platform?
I can answer this one, Singer. So we're aiming to make available high-quality, tested, error-proof tools. Hopefully that's the case. And we have slated, as I mentioned in the webinar, copy number and structural variant prediction using our published concerting and CREST algorithms. I also mentioned that we are going to produce a tool that can find mutational spectra from whole genome sequencing data. And there have been some really interesting insights from leukemia, for example. There's a signature of UV light-induced DNA damage in B-cell leukemias that we don't really understand why, but you can find it using this mutational signatures tool. And the last one we have in development is something called cis-X, we're calling it. You can find allele-specific expression. So if one allele is being expressed more than the other, and you've also found a variant that's maybe in the promoter, that might be evidence that the promoter is driving monoallelic expression of that particular gene. And so all of these are quite interesting areas that we're working in, and we hope to make those tools available to everybody. Not just to use them, but also to improve them and give us feedback.
All right, sounds great. Our second question is, what are the costs associated with running analysis on St. Jude Cloud?
I guess I'll take this one. Okay, so in terms of the cost, right, I know Scott already covered that all of the St. Jude data, the close to a petabyte of data, has been sponsored by Microsoft. So you can get a copy of that data for free. If you upload your own data to St. Jude Cloud, obviously there's a storage cost associated with that. So that costs some money, but it's relatively negligible, especially when compared to, the cost of storing things locally and investing in hardware, things like that. With the tools, it's a similar situation, right? So it costs money to run the tools because there's underlying computing infrastructure there, but that cost is pretty negligible. And we hope that, moving forward, labs will start building in the cost of running these tools in the cloud into their yearly budgets. There is one exception, though, for the tools. So the Pecan Pi variant annotation tool, you can just upload a VCF and St. Jude will pay for that. So I think that's all of the things that cost money in St. Jude Cloud. Scott, can you think of any others?
Well, I have one thing to add. Clay's got a team of software engineers who's trying to make the tools as efficient as possible, so to minimize the cost for the users. I think that's
right.
all I want to add.
All right. Then we have another question of how can I learn to use the St. Jude tools?
That's a good question. I'll take this one too, Scott, if you don't mind. So when me and my team are working with the biologists here and we're developing these tools and optimizing them, one of the things that we place a lot of emphasis on is making sure that it's very clear how to run them. What the results are spitting out is very clear because, we've had a lot of experience with bioinformatics tools being black box tools. So on our documentation site, we have a full guide, a full write-up from the authors of the tools and my engineers talking about how do you run this tool, how do you get your data in there, what are the different parameters that you can input, what are the results files that come out, and some gotchas about how you might interpret those results, and also like known issues, things like that. So we hope that that's where people are going to learn how to run our tools and what our tools do. But as always, we're trying to constantly make improvements to that. So you can always reach out to us, stjude.cloud/contact, and ask questions, and we'll also be updating documentation frequently.
All right. It looks like we have time for one more question. Any questions we haven't had time for, we will be following up via email. So for the last question, isn't this just DNA Nexus with a makeover?
Okay, I can answer this one. My answer would be no, it isn't. St. Jude Cloud sits on top of DNA Nexus, and we're really happy with that arrangement because the DNA Nexus toolkit gives us security, file management, workflow management, all things that scientists like me don't care about, but really have to care about. And DNA Nexus does a great job of that. All of our stuff that we've shown is exclusive to St. Jude Cloud. You have to go to stjude.cloud to access our workflows and our visualizations. Now, the other part of the story is, however, we don't suppose that there's going to be one cloud platform to rule them all. More likely, there's going to be an evolving cloud ecosystem with all of these sites talking to each other. And the really interesting thing is that DNA Nexus provides that ecosystem. There are other places like Mosaic Consortium, Precision FDA, also on DNA Nexus, and tools can move relatively freely between those subsites. So I've said that we have our St. Jude tools that are available through St. Jude Cloud. But what I didn't say is that there are a whole load of other tools available through DNA Nexus platform to do many, many things that we haven't had time to implement. Some of the new variant callers, for example, are available through DNA Nexus, and you can run them directly on St. Jude Cloud data. So hopefully that answers your question.
All right, sounds good. So we're about out of time now. I'd like to once again thank the team from St. Jude, both Scott and Clay, for their presentation. Any final words from either of you?
No, thanks very much for joining us.
Yeah, thanks for having us.
All right. Thank you once again, Scott and Clay. If you would like any more information about how to access the St. Jude Cloud Platform's public repository, or how to utilize the collection of bioinformatics tools and unique visualizations, email us directly at stjude.cloud/contact. If you're interested in building a community portal like St. Jude Cloud for your own research team, please email info@dnanexus.com or visit go.dnanexus.com/community-portals for more information. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing until April of 2019. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Enjoy.